The overall goal of this procedure is to produce a recombinant protein that is composed of a mixture of ordered and disordered domains for hybrid structural analysis by both X-ray crystallography and small-angle X-ray scattering.
This method can help to answer key questions in structural biology field about protein biological function through the determination of overall solution state structure of protein.
The main advantage of this technique is that it can be used to model dynamic regions of a protein or macromolecule that cannot be resolved by X-ray crystallography.
Generally, individuals new to this method will struggle at the beginning with learning how to properly assess the quality of their SAXS data.
To begin the procedure, transform an Nsa1 expression plasmid into a suitable E. coli expression strain. Culture the cells, induce Nsa1 expression, and harvest the cells. Resuspend the cells in 25 milliliters of lysis buffer, pre-chilled to 4 degrees Celsius and containing 1 EDTA-free protease inhibitor tablet. Sonicate the cells at 4 degrees Celsius for 7 minutes with a cycle of 2 seconds on, 2 seconds off. Then, centrifuge the lysate at 26,900 times gravity for 45 minutes at 4 degrees Celsius. Ensure that the mixture does not freeze during centrifugation. Next, equilibrate a gravity flow column containing 10 milliliters of immobilized cobalt affinity resin with lysis buffer. Load the clarified lysate onto the column and allow it to flow through the resin at 4 degrees Celsius. Then, wash the resin twice.